So now I'm going to click your forward one, click your back one. It should be good to go.
Hi, I'm Annie Down. This is my presentation on the mechanism of mitochondrial localization of the steroidogenic acute regulatory protein. So, the abstract of my project here is to study the mitochondria of the cell in detail and observe steroidogenesis in an effort to determine where in the cell the steroidogenic acute regulatory protein is. The STAR protein is steroidogenic acute regulatory protein for short. Is essential for steroidogenesis and is involved in the transport and binding of cholesterol. Not much is known about the STAR protein currently, as a disease is caused by a STAR mutation. As a disease that is caused by this mutation does not affect a large number of people, but the current model in the field is that the STAR protein is located in the mitochondria, but where specifically in the mitochondria is yet to be determined. So what exactly are mitochondria? They're endosymbiotic organisms that were once free-living prokaryotes that were engulfed by a eukaryotic cell and developed a symbiotic relationship, and now it permanently lives as part of the eukaryotic cell. And its functions are energy production as well as steroidogenesis, which will be the main focus. So this is what the mitochondria looks like in simply. So you have the outer mitochondrial membrane, and you have the inner mitochondrial membrane right here. In between the two membranes is the intermembrane space, and inside the inner mitochondrial membrane is the matrix. So steroidogenesis is the synthesis of steroid hormones. The four steroid hormones are testosterone, estrogen, progesterone, and corticosterone. And all of these four hormones are made from pregnenolone, and pregnenolone is made in the matrix of the mitochondria. The most important step for steroidogenesis to occur is for cholesterol to enter the mitochondria, and the cholesterol goes to the matrix where it becomes pregnenolone, which then makes the steroid hormones. So let's look a little bit at the STAR protein. It was identified by Dr. Douglas Sacco at Texas Tech. It's essential for steroidogenesis, and it binds and transports cholesterol. So the current knowledge we have about STAR is that it is only expressed when hormones are being produced. There were experiments about STAR knockout mice. Knockout mice is when you produce mice that don't express STAR so that you can compare it with wild human mice. So these knockout mice cannot produce testosterone, estrogen, progesterone, and corticosterone because they don't have STAR. STAR mutations in humans cause a disease called congenital lipoid adrenal hyperplasia, or abbreviated CLAH. If you look at the panels, I guess, panel C shows what a wild-type mouse, the tissue section of an adrenal gland of a wild-type mouse, and panel D shows a tissue section of an adrenal gland from a STAR knockout mouse. Let me explain it a little here. So what's going on is that, hold on, let me backtrack a little bit to the congenital lipoid adrenal hyperplasia disease. Congenital means that you were born with it. Lipoid means that CLH causes fatty deposits. Adrenal is of or relating to the adrenal gland, and hyperplasia means an increase in size, generally referring to a gland. So there are several symptoms of this disease here. So there are low levels of steroid hormones when born. Adrenals are enlarged, fatty deposits in the adrenal gland, and there's a lack of testosterone, causes the patient to be born with axial female genitalia. So, backtracking to the pictures, this is a wild-type mouse, which means they express STAR normally. So this is the adrenal gland, and the red parts are the fatty deposits. And since STAR isn't there to bring the mitochondria in or out, the fat gets deposited here and it doesn't go out, which causes the adrenal gland to swell. So CLH can be treated with hormone supplements and cosmetic surgery. It's caused by the mutation in the STAR gene. But the mechanism behind STAR's ability to transport cholesterol is unknown. So we do know that STARs affects CLH. We don't know exactly how. So the current work is that steroidogenesis occurs in the mitochondria. STAR is essential for steroidogenesis, but we still don't know the exact localization. And the model in the field that's currently being tested is that STAR is located in the mitochondria. So this is an experiment that my sister did in the lab, where she took Lidotracker Green, which is I think it's a it's just a green tracker, and she put them on the mitochondria of kidney cells, and then she took Star Apple, which I think binds to the STAR protein, and then she looked at the two merged here, and you can see the two are practically on top of each other, which means that STAR should be where a mitochondria is. So here are Leydig cells, and I believe Leydig cells, let me see. Leydig cells are cells that naturally make steroid hormones and express STAR naturally. So kidney cells don't naturally express STAR, but we use them because they're easy for scans and they're just the ideal shape for scans. But Leydig cells naturally express STAR, so it shows here that wherever there's a mitochondria, there will be a cell, more or less. So let me explain a little bit about mitochondrial targeting signals. All mitochondrial proteins have a mitochondrial targeting signal, which is shortened to MTS. MTS is a helix at the beginning of a protein, so it's this. And this sends the protein to the matrix. So some mitochondrial proteins have a second MTS, so we're talking about this part, and these proteins are sent to the IMS. So let me backtrack a little bit. So if it only has one section, it's going to go only to the outer mitochondrial membrane, but if it has two trackers, it's going to go to the inner mitochondrial membrane. Okay. So now that so determining the location of STAR. Once again, the STAR protein's function is still unknown. We don't know how it transports cholesterol, like how exactly, and we don't know the origin and the destination of cholesterol. So let me explain that. That means it could start anywhere in the cell. We don't exactly know where STAR comes into play from the process from the cell membrane to the matrix of mitochondria. And the future work cut out for me is going to be to analyze the sequence of the STAR protein for possible mitochondrial targeting signals, which are these things. And we test for MTS, and we test if it has a signal first. Then we test if it has one or two signals. And if it has two, we have to identify the beginning of where the first one ends and where the second one, where the second one begins, I guess. So the methods to do all of this is to extract RNA from the cells that express the STAR, amplify it using PCR, which is So PCR is a method where you take the RNA and then you put it into a plasmid, and then you put the plasmid in bacteria, and then you multiply it to get more. To make mutated versions of STAR and then place in the plasmid, precise changes can be made to STAR sequence without restriction cloning. Then transfect the plasmid into the STAR knockout MA10 cells, which are the Leydig cells that were in the diagram earlier. They're the starogenic cell line, which means they naturally express STAR. STAR is removed from the cells using the CRISPR-Cas9. And then we use confocal microscopy to get an image to see if the mutated STAR can still localize to the mitochondria. So you see if in the diagram if star still appears where the mitochondria appear. And then you use hormone assays to determine if star is localized to the right compartment of the mitochondria. So that way you can determine which compartment the star is in. And here's my assay.
Questions? Remember, I'm keeping track.
Can you go back to the slide with like the images? I think you said like, because I'm just confused. You said the kidney cells don't express the star,
Yeah, they don't naturally express star.
so that the cells that you got, how are they expressed?
You just insert into the kidney cells.
What was that?
You just insert the star into the kidney cells.
And then like, so they're just inserted into
Yeah.
the cell.
They don't naturally express stars, so you have to put them in the cell.
You're
Not like putting them in the mitochondria, you're putting them in the cell. And then the cell does whatever it does to get star.
I think the question is, are you genetically modifying the kidney cell to express star?
Yes.
Or are you literally putting star protein into the cell? Most likely you're genetically modifying it. Is that the question?
Yeah.
Good question.
So if you know that stars wherever mitochondria is,
Yeah.
what's like the location to be determined still?
Oh, so we know it's in the mitochondria, but we don't know exactly where. And based on where it is, will also determine what its role will be. So where it like comes into play in the process of bringing cholesterol to the matrix. Steroid hormones. Okay. So hold on, I don't know the whole thing. Okay, so so all of these the main four hormones that steroidogenesis produces are essential to life. So testosterone, estrogen, obviously are made for reproduction. Progesterone, for purposes of this class, is basically works like estrogen, and corticosterone kind of works with like your thyroid gland, I believe. So that's like essential to life. If you don't have it, firstly you're gonna have if you back up, forward. If you don't have it, then you essentially have CLH.
The genes expressed in steroidogenesis, are they expressed in all mitochondria and all the specific ones?
Yeah.
Can you say that one? I didn't hear the question. Just say it a little bit louder.
So like are genes that express steroidogenesis, are they expressed in all mitochondria throughout the body or just
Yeah.
specific ones?
Because the mitochondria is known for energy production, but it also makes steroidogenesis. So every single mitochondria also does steroidogenesis.
Is this specific?
It's every single one.
But there are some cells, like skin cells, that do produce steroids as well. So do they express this gene as well?
That's a good question.
It's
Probably
okay.
not.
It's okay not to know something. You don't have to be 100% on everything. If you ask a question that you're not sure of, you could just say.
I'm pretty sure they don't actually. It's probably just things that actually exist on us. Wild type field mouse gas. Haha. Does anyone have questions?
Any other questions? So can you go to the targeting slide? Right here. Do you have any examples of molecules, whether they're other hormone molecules or other proteins that display these targeting signals that you're looking for? So basically, we know about these targeting signals, and
Yeah.
we're expecting to find these targeting signals in the star protein.
Yeah.
And if we find them, in the star protein, if we find them in the star protein, then we'll have a good idea of where they're located.
Yeah, we'll have an idea where it's going to be in the mitochondria.
Exactly. So my question is, do you have any example of proteins that they looked at that they found these targeting symbols in?
I'm pretty much sure like every common protein that's like well said that goes to the mitochondria.
Right.
I don't have a specific example, but
Okay.
this is like a pretty well known thing.
Okay. That's fine. And then when you go to your methods again, your extract RNA, express star, amplify RNA, make mutated versions of star. Do you know what mutated versions you're looking at? Like what you're
You just probably make knockout versions. Yeah. Or no, what you do is so star is a certain length,
Yeah.
but we're not sure because a certain length also involves the targeting signals.
Okay.
So we're not sure where the targeting signals like if they even exist. So we're looking at the protein part of it,
Yeah.
and we're just gonna mutate it to chop off parts of the protein thing to see if it still works and to see
Okay.
if it still appears in the mitochondria.
Okay, so right now you don't have any specific region that you're going to knock out. You're not
Yeah,
looking at a
it's just
specific
gonna
region.
be like-
You're just going to kind of randomly
Yeah.
knock some places out, and with the hopes of finding a location that it's still functional, I guess, and just not quite
Yeah, it's like where it
where it's
still
going to end up
appears.
located in the mitochondria.
So I talked about CLAH, which is right, a dysfunction that happens with respect to like the gene. You said that it's caused by a mutation in the star genes. Do we know what specific mutations, like what gene changes lead to?
It just means you don't produce star. So if star isn't there to bring out the cholesterol, then you're adrenal-glycosyls. And you're going to flow.
Do you know what specific genes like lead to?
Knockout mice live without their star protein.
No, they die within like 10
Okay.
Yeah,
So
because humans can get it
within what
treated.
time? Did you say 10 minutes?
Yeah.
Okay. Alright. So just go back to that slide again. Go back one more. To the one with the cross sections of the mice tissue,
Oh,
showing the cholesterol. This one. So this is of a fetal mouse, I would assume.
yeah. And that is like really
Any questions?
Human disease
Oh.
lethal?
It is if it's untreated. If you treat it, you can treat it with hormone supplements because they can't produce their own hormones. So you just inject them with hormones every once in a while, and they'll be okay.
When you're saying lack of testosterone causes external phenylketonuria, does that mean that on the inside there's a lack of it, or are there more to it?
I don't know the specific details, but I'm pretty sure you're born with phenylketonuria, and then you're born with both if you're supposed to be male. So that's.
Could you repeat that question again? What was that?
I was asking if the lack of testosterone, since it causes the external phenylketonuria, if it caused like something there's a lack of internal, or as
I'm pretty sure it's just the whole thing, but like you said.
So, are you trying to like correlate expression of SAR with like development of CLAH, like no expression of SAR? Because
We already know that CLH is associated with STAR. It's just an example of a disease because STAR isn't well studied because this can be easily treated. So this is just caused by STAR as an example. It's not like too vital to STAR, but we're trying to mostly just research STAR in general and see like what it does and everything. So it doesn't have much to do with CRH.
And CLH only be caused by mutation in the star gene. It's like the only.
Could you say that louder?
Oh, is CLAH the only can it only be caused by the mutation in the star gene?
Pretty sure.
It's like the only
Yeah.
it's a mutation from birth.
Hmm. Oh yeah, you can't develop that.